Bismillahirrahmanirrahim. In this video, we are going to discuss the historical perspective regarding repair, regeneration, and fibrosis. The process of tissue repair, regeneration, and fibrosis are fundamental to the survival of organism, and these mechanisms enabling them to recover from injury and disease. So, whenever there is a damaged tissue, it heals for the survival. So these understanding of these mechanism has evolved significantly, and it is shaped by observation, experiment, and discoveries from ancient times to modern era. Throughout history, scholars and scientists have sought to understand how the body heals and regenerates damaged tissue. The evolution of our understanding reflects not only the growth of scientific knowledge but also the influence of cultural, philosophical, and technological context in that particular era. In ancient Greece and Egypt, uh, evidence of clotting of the blood was found in the literature, as stated by Edwin Smith Papyrus in 1600 BC, and his papers, his writings, and other evidences, they have got reference for wound healing and surgical practice that was implicated in that era. And it was also known that they deputed surgeons during war to manage the wounds. Only the advent or invention of the microscope revealed the implication of some cellular theories that we are going to see later. Before that, there was no understanding about the cell and their interaction in the process of repair, regeneration, and fibrosis. Hippocrates during 460 to 370 BC, he was considered father of Western medicine. He postulated that this healing was a natural process, and he termed this as vis medicatrix naturae, the healing power of the nature. He designated that this is the healing power of the nature that heals the wound. He described the inflammation and suppuration as part of the healing process, and also hinted at early clinical insight into wound repair. He said that as there was a predominant theory of humoral theory or fluid theory that was predominantly accepted during that area, he said if the pus is white and not offensive, healing will follow. But if it is sinuous and muddy, that then death is to be looked for. Galen in 129 to 200, who was a Roman physician, expanded upon his Hippocratic ideas, and he stated that healing occurs through the process of concoction and reconstitution, which involves the transformation of blood into a new tissue. He also introduced the term of laudable pus, pus bonum et laudabile. So this theory was accepted for more than 1,000 years, both the Western and all over the world. So So he also emphasized it is important to the pus then that this pus, if it is present and if not drained, could prove fatal. Later in 1267 and 1321, the Theodoric Borgogni and Mondeville, they questioned his idea of the laudable pus, and they said even if the pus is there and if it is white, it is also associated with morbidity and mortality. But despite this, the belief of this laudable pus persisted until the 19th century. Then there was an empirical shift in this theory. In 1880, the dominant system in the United States was the spoils system within the government, particularly in local and state politics. The system involved distribution of the government jobs and contract as a reward to loyal supporters of the winning political party. So James Garfield, who was the 20th president of US, he was the first who discontinued this spoil system. He tried to invoke a system based on merit. So he was tried to be assassinated, and his assassin Vito, who was his supporter during election campaign, believed he deserved a diplomatic post in Europe for the support of Garfield election, but Garfield refused to appoint him. After bullet injuries, the Garfield survived the bullet injuries, but he died of sepsis month after the shots. And this was later, when inquired, this was this death was due to infection from repeated probing by non-sterile instruments. So this highlighted the importance of antiseptic techniques and dangers of ignoring the germ theory that was proposed by Joseph Lister against Semmelweis and Louis Pasteur in 1860s and 70s. The Joseph Lister, he introduced antiseptic surgical techniques using carbolic acid that drastically reduced infection rates, especially in post-surgical wounds. So his theory was widely accepted, especially after that incident, and his measures also improved the healing outcome. Then there was James Lind, who described the role of collagen and vitamin C in the healing process and ran a clinical trial for scurvy by use of lemon and orange. Then Theodore Froelich and Axel Holst, they also insisted on the necessity of ascorbic acid for the action of propyl hydroxylase for proper folding and stabilization of the collagen helix. So during this period, Vesalius, who was an anatomist, he challenged the Galen's theory and still remember that microscopic world was hidden. So microscope was invented in 17th century that changed this. these theories. Leeuwenhoek made various microscopic observations and during 18th and 19th century, and these advances in histology, they began to shed light on cellular composition of the tissue. And the Sir Rudolf Virchow, who is the pioneer of the cellular pathology, and he also emphasized that all the disease processes, including healing, fibrosis, and others, they originate at the level of the cell. And he also introduced the concept of chronic inflammation. He also recognized the proliferation of the connective tissue in the pathological repair processes. The concept of regeneration was lying there since the ancient Greek time. The Prometheus was punished by the Zeus, and a bird daily came to eat his liver that generated by the evening. So there was penetrating body's healing power the people were aware of. So liver is one of the few human organs with true regenerative capacity. In the early 19th and 20th century, there was emergence of concepts of regeneration. Scientists began to distinguish regeneration from mere healing by scarring. The experimental studies on invertebrates or amphibians such as limb regrowth in salamanders revealed that the capacity of some organisms to regenerate entire body parts, a phenomenon not mirrored in the mammals. The Wilhelm Rocks and August Weismann debated whether this regenerative potential was a retained ancestral trait or regulated biological capacity. The work of Thomas Hunt Morgan in during 1866 to 1945 helped to establish the principles of tissue polarity and cellular memory that formed a foundational understanding of regenerative biology. So continuing this regeneration concept, Sir John Gurdon in 1950 showed that differentiated cell could give rise to entire organism, proving cell plasticity. This concept paved the way for stem cell therapy in regenerative medicine, including the development of induced pluripotent stem cells in the 21st century. Then the mid 20th century was the molecular era. In molecular era, there was a rise of molecular biology. Tissue repair began to explode at the level of the genes, protein, and signaling pathway. There was discovery of growth factors such as epidermal growth factor and platelet-derived growth factors that provide mechanistic insight into how cells communicate during wound healing and regeneration. Then there was studies about the fibrosis. So this fibrosis study gained prominence, and particularly pulmonary and liver fibrosis or liver cirrhosis and heart failure. There were many studies on these conditions. So fibrosis was recognized as the maladaptive result of excessive extracellular matrix deposition and the transforming growth factor beta signaling and myofibroblast activation were considered responsible for these errors or these malformations. Now there are contemporary advances in the uh, tissue regeneration and tissue healing, and there uh, the scientists have now contrasted very well between regeneration and fibrosis. Many key discoveries have been done in the modern era. One is the plasticity of the stem and progenitor cell in the tissue like intestine, skin, and liver. Then role of macrophage and transformation and differences between M1 or classical macrophage and alternate macrophage or M2 that determine the outcome of the chronic inflammation. Then there is identification of fibrogenic pathways such as transforming growth factor beta, SMAD axis. Then there is extensive study of the influence of the immune system, mechanical stress, and epigenetic regulation in determining the healing outcome. Then there are many advances in the modern era in the biomaterials, tissue engineering, and gene therapy that are offering prospects of promoting regenerative healing and limiting fibrosis. So experimental therapies such as antifibrotic agent, stem cell transplantation, reprogramming of the myelofibroblast are being tested in various clinical settings. Now the scientists have knowledge that if there is injury, it can be healed by regeneration or fibrosis. If there there is fibrosis, it will also accompany the loss of function. After the fibroblast activation, there is deposition of collagen and the normal tissue is lost over here. But if they can regenerate the tissue, there will be normal restoration of the tissue structure as well as function that is due to proliferation and differentiation of the activated resident stem cell present in that particular milieu. So the understanding of tissue repair, regeneration, and fibrosis reflects the trajectory of biomedical science itself from mystical speculation to empirical observation and finally to dissection of the molecule. Ancient physicians, they recognized the body's remarkable ability to heal, but the modern medicine now seek not just to aid recovery, but now the medicine is guiding the tissue regeneration and preventing fibrotic complication. As the research advances, the boundary between scarring and regeneration continues to blur, and there is hope for regenerative therapies that mimic the nature's most elegant solutions. Thank you for watching this video. If you have any question, please write in the comment box.